Welcome to Section 1 of Laboratory Techniques. In this section, we'll be discussing polymerase chain reaction, or PCR. Let's get started. PCR is a molecular lab technique that amplifies DNA. So it's a method of amplifying DNA. There are 4 elements which are necessary in order to perform PCR, and PCR occurs in 3 steps. The 4 elements include a source DNA template, knowledge of the adjacent regions to the target that will be amplified, a DNA polymerase that can withstand high temperatures, and deoxynucleotide triphosphates. Let's pull up a figure so you can see what I'm talking about. This is a detailed figure of PCR which can be found in Section 1 of Lab Techniques. The DNA, which you can see right here at the top of the image, is shown as it's being amplified, and the bottom part of the image shows a graph of the cycle time and temperature. Notice cycle time right here and temperature right here. So we have the source DNA template at the top of the image, and for example, this could be a region of chromosome 21 to investigate Down syndrome, a region next to the CFTR gene to investigate cystic fibrosis, or any other region in the genome. You get the idea. We need a source of DNA in order to amplify a region of interest. So element number 1 is a source DNA template. Next, we need to know the exact sequence of the flanking sequences to the target that will be amplified. Notice that the flanking sequences are shown right here. You can see the nucleotides on the bottom strand of the left flanking sequence right here, which are GATC. With this information, we can design a primer which is complementary to this region. Notice that the primer, which is shown later on right here, is complementary to the flanking sequence. So G pairs with C, A pairs with T, T pairs with A, and C pairs with G. So again, the second element necessary for PCR is knowledge of the flanking sequences to the target that will be amplified. The third element necessary for PCR is DNA polymerase. You can see DNA polymerase right here. Finally, the fourth necessary element is deoxynucleotide triphosphates. You can see that deoxynucleotide phosphate bases are shown right here. Okay, now let's cover the 3 steps of PCR. Once all of these elements are combined into a tube, the sample can be amplified. This is done by heating the sample, also known as DNA denaturation. You can see on the graph that the temperature increases right here, and that this represents DNA denaturation. Notice that above the graph, the DNA is being separated into 2 separate daughter strands, right here and right here. This is the first step in PCR. Next, the sample is cooled, which allows the primers to anneal. You can see on the graph that the temperature drops right here, and above the graph, notice that the primers are annealing right here. This is the second step in PCR. Finally, the sample is warmed, and DNA polymerase copies the DNA. Notice on the graph that the temperature rises right here, and notice that above the graph, the DNA is adding nucleotides right here. This is also known as extension or elongation, and is the third step in PCR. From your text, you can see the 3 steps listed here. As we just discussed, these include denaturing of the DNA, annealing of the primers, and elongation by DNA polymerase. Okay, one more point before we move on. Once the DNA is amplified, the sample can be transferred to a gel plate to undergo electrophoresis. The intensity of the band on a gel can then be used to provide semi-quantitative results. This figure shows bands that represent DNA from gel electrophoresis. This can be found in Section 1 of Lab Techniques. Notice that there are several bands seen in this image. For example, this is a band, and these all are bands shown on this column right here. Each of the bands represent an amplified region of DNA. If the strand is brighter, it means that more DNA is present. So in this way, traditional PCR can be used to provide semi-quantitative results. In a second, we'll discuss an alternative way of performing PCR, which can provide quantitative results. Okay, let's do a question. A 37-year-old female has been working with an infertility specialist in an attempt to become pregnant. During in vitro fertilization, an egg is retrieved from the ovaries. The egg is then artificially inseminated and prepared for uterine implantation. However, the patient requests that the embryo be screened for genetic mutations prior to uterine implantation. A sample of DNA is obtained from the embryo, and PCR is performed to screen for several genetic abnormalities, including Huntington disease. A gene on chromosome 4 is amplified, and the embryo produces a PCR product that is much larger than expected. What does the large PCR product most likely indicate about the embryo? Hopefully from the question stem, you notice that a gene on chromosome 4 was amplified by PCR in order to screen for Huntington disease. Recall from genetics that Huntington disease is an autosomal dominant trinucleotide repeat disorder. This means that patients with Huntington disease will have repeated sequences of CAG found on chromosome 4, so something like this. From the question stem, hopefully you notice that when this region was amplified, it resulted in a product that was much larger than expected. In other words, there must have been CAG repeats on chromosome 4, which resulted in a larger than expected PCR product. Therefore, we can conclude that the large PCR product most likely indicates that the embryo has Huntington disease. Okay, let's move on. Real-time PCR is another type of PCR that can be used to amplify DNA. In this type of PCR, the method I described earlier with traditional PCR is also used. However, a single-stranded DNA fluorescent probe is also used. When DNA polymerase replicates the DNA, the probe is removed and becomes activated, resulting in light emission. The fluorescence intensity can then be used to quantify the DNA. In this way, real-time PCR can be used to provide quantitative results. As you'll see in a second, the curve which crosses the threshold line first had the highest concentration of DNA in the initial sample. Let's pull up a figure so you can see what I'm talking about. This is a figure of real-time PCR which can be found in section 1 of lab techniques. Notice that the x-axis represents the rounds of PCR and the y-axis represents the fluorescence intensity. Also notice that this dotted line right here represents when the sample can be detected, so the threshold of detection. So for example, let's assume that we have 3 separate samples of DNA. Each sample is being amplified at the same time. Because this line right here crossed the threshold line first, we can conclude that it initially contained the most DNA. This makes sense, right? If a sample starts with 100 copies, then it would be amplified faster than a sample that only started with 1 copy. Okay, now let's discuss reverse transcription PCR. Reverse transcription PCR is exactly what it sounds like. We're amplifying mRNA into DNA, so the reverse of transcription. This is done through the enzyme reverse transcriptase. So a template of cDNA is produced from mRNA by reverse transcriptase. This is performed very similarly to traditional PCR and real-time PCR. A sample of mRNA is heated up to linearize the molecule of mRNA. Deoxyprimers anneal, and then reverse transcriptase converts a segment of mRNA into cDNA. It's important to note that complementary DNA, or cDNA, doesn't contain introns because it's made from mRNA. Recall from molecular biology that the introns of mRNA are removed during processing, so mature mRNA lacks introns. Finally, you should know that by combining reverse transcription PCR with real-time PCR, we can actually quantify the amount of mRNA in a cell, so it can be useful in measuring mRNA. This has many applications, including the ability to measure the treatment response of certain cancers. Let's do a question to illustrate this point. A 14-year-old boy is diagnosed with acute lymphoblastic leukemia. He is started on an aggressive chemotherapeutic regimen and receives regular follow-up care. During one of these visits, his treatment response is analyzed by using reverse transcription PCR. A bone marrow aspirate is obtained, and mRNA created by a BCR-ABL translocation is then used as the template for real-time PCR. How will the PCR product most likely differ from the BCR-ABL gene? Hopefully from the question stem you notice that this boy has acute lymphoblastic leukemia and that his treatment response is being monitored by using reverse transcription PCR followed by real-time PCR. Let me explain how this is done. BCR-ABL is usually associated with chronic myelogenous leukemia, or CML. However, sometimes it can be present in ALL, and the mRNA produced by BCR-ABL can then be used to measure the treatment response. For example, if the chemo drugs are effective, then the mRNA produced by BCR-ABL will decrease. So if the drugs are effective, then the mRNA will decrease. If the drugs are ineffective, then BCR-ABL will be very active, resulting in a lot of mRNA. So if the drugs are ineffective, then mRNA will increase. In order to quantify the amount of mRNA, we first need to convert the sample into DNA so it can be used in real-time PCR. So mRNA is converted into cDNA. From here, the cDNA can be amplified and quantified by using real-time PCR. As you can imagine, the quantitative results of real-time PCR provide incredibly valuable information because they tell us how much mRNA the cancer is producing. Because mRNA lacks introns, the cDNA PCR product will also lack introns. So in answer to the question, the PCR product will most likely lack introns, which is how it will differ from the BCR-ABL gene.